Good day, dear students. And today the theme of our lecture, allergy, allergic disease for future practitioners. Allergic disease are a major health problem across the world. And when we talk about disease in the 21st century, we talk about epidemic of allergic disease. The global prevalence of allergic disease increased and affected from 10 to 30% of global population. Apart from health problem, allergic diseases are also a major social, economical burden of a country. The prevalence of allergic disease increased in the last 20 years. Approximately 30 to 40% of the world population suffered from different allergic disease. Estimated 300 million individuals worldwide have bronchial asthma. And we think that prevalence of asthma increased to 400 million by the 2025. Allergic rhinitis also it's very important allergic disease. Allergic rhinitis is a risk factor for asthma, and allergic rhinitis affected about 400 million people annually. Also very important allergic disease is food allergy. Food allergy affected from 200 to 2.5 million people. And when we talk about Europe, about 150 million of Europe citizens suffering from chronic allergic disease. And to 2025, more than 50% of all population will suffer from allergy. And when we talk about diagnosis of allergic disease, on this picture, you can see that about 45% of allergy patients are likely to be misdiagnosed in Europe. When we talk about different type of allergic disease, about 100 million Europeans suffer from allergic rhinitis, and 70 million suffer from asthma. Asthma and allergic rhinitis alone are estimated to result with more than 100 million lost work days and missed school days every year. And also you can see costs treatment of patients with asthma and allergic disease. Prevalence of allergic disease. The prevalence and complexity of allergic disease also increased, especially among children and young adults. Major allergic disease, as you can see in this picture, it's eczema. You see increase in the first year of life. Also very important. Recurrent wheeze, bronchial asthma that also increase. Also you see increasing of rhinitis and food allergy. When we talk about allergic rhinitis, but it is a very important disease because first of all we see comorbidities of allergic rhinitis and asthma, allergic rhinitis and atopic dermatitis, rhinitis and conjunctivitis, and sinusitis, polyposis, and some other disease. And secondary, when we talk about allergic rhinitis, we think that allergic rhinitis decreased quality of life. also lead to sleeping disorders, learning and attention impairment. And also in patients with allergic rhinitis, we see mouth breathing, dental, and others problem. And on this picture, you see a world map of prevalence of diagnosis asthma and proportion of population. You see red, more than 10% of population have bronchial asthma, and yellow from 2 to 5% of population have bronchial asthma. And also you see white and blue color on this picture. In this country, we don't know about prevalence of bronchial asthma. Prevalence of food allergy. In some countries, we see that about 10% of population affected of food allergy, 10% of population affected of drug allergy, and 20% of patients suffering from other form of allergy. When we talk about food allergy, it's very often, as you see in this picture, we see in children. When we talk about drug allergy, very important that among drug allergy, penicillin becomes the most common drug leading to a hypersensitivity reaction. And on this picture, you see that in the last year, we see decrease percent patient with infection disease, decrease patient with tuberculosis, hepatitis A, rheumatoid fever, measles, and some others, and increase prevalence of patients with allergic and autoimmune disease. You see increased percent of patients with diabetes and bronchial asthma. Socioeconomical impact of allergic disease, also very important. For most people with respiratory allergies, their conditions significantly reduce their quality of life, affecting both work and leisure time. They can relieve their symptoms by trying to avoid allergen exposure, but restricting their lives in this way can have physiological and social consequences as well as socio-economical impact. According to a European survey of diagnosed allergy sufferers, around 80% of respondents said their condition considerably affect their daily activities. The increasing prevalence of allergy also has major economical consequences for society through reduced working capacity and more sick leaves. And some words about history of development science about allergic disease. First description of allergy belong to description disease of king Menses of Egypt. This king was killed by the sting of a wasp, and it was sometime between 3000 and 3,300 BC. Also, we know about Britannicus, the son of the Roman Imperator Claudius, and Britannicus have allergic rhinitis, and Britannicus has allergic to horses. And Britannicus developed a rash and his eyes swelled to the extent that he could not see where he was going. Also, a very important history of King Richard III. King Richard III have allergic reaction to strawberries, and clinical manifestation was development acute urticaria. And King Richard usually ate some strawberries just prior to given audience. And also King ate strawberries before audience to Hastings, it's a lord. And King developed acute urticaria. He then accused Hastings of putting a curse of him, and action that demanded the head of Hastings on a plate. And Roman philosopher Lucretius write about food allergy. What is food for some may be versus poison for others. And modern history of allergy and allergic disease, described of allergic disease, belong to Dr. John Bostock. He first described high fever in 1819 as a disease that affected the upper respiratory tract. And now we knew high fever as allergic rhinitis. Common symptoms include sneezing, running and stuffy nose, red, itchy, swollen eyes, and itching in the nose and throat. Charles Blakely, also very important in allergic history, because he first performed skin test, and it was in 1869. And now we also use skin test for diagnosis patient with allergy. Blakely found a positive reaction to a specific allergen becomes evidence in about 20 minutes by the appearance of a hive-like response at the tasted skin site. In 1902, Charles Richt and Paul Patier invented the word anaphylaxis. And also, we use this word in our days. This scientist, in the course of immunization, they discovered life-threatening response to medication and protein substances. And it often occurs in people who are particularly sensitive to penicillin or maybe other antibiotics, stinging insect, shellfish, peanuts, tree nuts. And anaphylaxis, may be life-threatening reaction, and we must treat it immediately. On this slide, you see photo of Clement von Perkier that in 1906 first used the word allergy. He described strange non-disease related symptoms that some diphtheria patients developed when treated with horse serum antitoxin. And the word comes from Greek word elos, meaning change in the origin state. In 1914, the work of Leonard Nunn and John Freeman helped establish the basis of immunotherapy or allergy shots. And also we use immunotherapy for treat our patient in our days. We can use different type of immunotherapy. Very important, sublingual immunotherapy. It's very useful for our patient, and we see a very good effect after using this type of immunotherapy. In 1937, Daniel Bowett synthesized the first antihistamine drug. And this was a first beginning of first generation of antihistamine drug. Now we have two generation of antihistamine drug. We have different antihistamine drug, more than 200 of antihistamine drug. And also we use antihistamine to block effect of histamine and protect our patient from development of some symptoms of allergic reaction. In 1948, Philip Hench and Edward Kendall discovered and introduced corticosteroids into clinical medicine. And it is very important because, we use systemic corticosteroid for treat our patient, and also very important that we use topical corticosteroid. Especially it's very important for patient with asthma and allergic rhinitis when we use corticosteroids long period. In 1953, James Lee and Jeffrey West discovered the mast cell granule to be the major source of histamine in the body. Lee and West dependent on a value partner and experimental subject dog named Judy. This cocker spaniel earned a place in canine history thanks to her mast cell tumor, which had the highest histamine content ever recorded. In 1976, Kimishit and Teruka Ishizaka further explained the allergy process by discovering the role of immunoglobulin E class antibodies as a principal mediator in allergic reaction. And we talk about total IgE and specific IgE, and immunoglobulin E are very important in development immediate type of allergic reaction. In 1980s, Bent and Samuelson received the Nobel Prize in Medicine and Physiology for identifying leukotrienes as the slowing reactant substance of anaphylaxis. And now we talk about some terms that we can use in our classes. First, we talk about hypersensitivity. Hypersensitivity causes objectively responsible symptoms and signs initiated by exposure to a defined stimulus that is tolerated by normal subject. And on this picture, you see classification of hypersensitivity. All type of hypersensitivity we divided into two groups: allergic hypersensitivity and non-allergic hypersensitivity. Allergic hypersensitivity are when immunological mechanism defined or strongly suspected. Allergic hypersensitivity divided into two groups: IgE-mediated or immediate type of allergic reaction or first type of allergic reaction, and non-IgE-mediated allergic hypersensitivity. Non-allergic hypersensitivity also we see in our patient. First of all, when we talk about food allergy, or sometimes when we see patient with drug allergy, in this case, immunological mechanism excluded. And next, we will talk about atopy. Atopy is a personal and family tendency, usually in childhood or adolescence, to become sensitized and produce immunoglobulin E antibodies in response to ordinary exposure to allergen. Usually, these allergen are proteins. The terms atopy and atopic should be received to describe the genetic predisposition to become IgE sensitized to allergens, commonly occurring in the environment and to which everyone is exposed, but to which the majority do not produce a prolonged IgE antibody response. So atopy is tendency become sensitized and produce immunoglobulin E. And some of these patients have clinical manifestation and some don't have clinical manifestation. Atopy is a clinical definition of immunoglobulin E antibody high responder. And the term atopy cannot be used until IgE sensitization has been documented by Ig antibodies in serum or by positive skin prick test, which also demonstrate Ig antibodies in patients. Allergic symptoms in atypical atopic individuals can be referred to as atopic. For example, it may be atopic asthma, atopic dermatitis, and other. Neither positive skin prick test nor presence of immunoglobulin E antibodies to a less common allergen, for example, immunotherapy or a drug, which a high dose exposure is a diagnostic criterion for atopy. And what is allergy? You remember that allergy is a hypersensitivity. It's a hypersensitivity reaction initiated by immunological mechanism. So when we talk about allergic reaction, we think about immunological mechanism in development of this type of reaction. When we talk about mechanism of allergy, allergy can be antibody mediated or cell mediated. Cell-mediated allergy is the late type of allergy. In the majority of cases, the antibody typically responsible for an allergic reaction belong to immunoglobulin E isotype. And these individuals may be referred to as suffering from IgE-mediated allergy. When we talk about IgE isotype of allergen, we think that development of allergic hypersensitivity on first type of allergic reaction or immediate type of allergic reaction or IgE type of reaction. In non-IgE mediated allergy, the antibody can belong to immunoglobulin G isotype. For example, sometimes you see patients with anaphylaxis due to immune complexes containing the extract. But in some group of disease, for example, allergic bronchial pulmonary aspergillosis, we see both immunoglobulins, immunoglobulin E and immunoglobulin G. Allergic contact dermatitis is delayed type of development allergic reaction, and this disease mediated by lymphocyte. First allergic reaction, mechanism of allergic reaction, described by Prausnitz and Küstner in 1921. And also you remember that only 45 years later, Ishizaka described immunoglobulin E. On this picture, you can see 4 types of hypersensitivity reaction. First type of hypersensitivity reaction, also be called IgE-mediated hypersensitivity. IgE-mediated hypersensitivity. And you see scheme of reaction on this picture. Typical manifestation includes systemic anaphylaxis, also high fever, asthma, food allergy, and eczema. Type 2 of allergic reaction, it's IgG-mediated cytotoxic hypersensitivity. As example of this group of reaction may be blood transfusion reaction, autoimmune hemolytic anemia, and some other reaction. Type 3, immune complex reaction. Manifestation include Arcturus reaction and generalized reactions such as serum sickness, necrotizing vasculitis, glomerulonephritis, and some others. Type 4, it's a cell-mediated hypersensitivity. And manifestation of this type of reaction include contact dermatitis, tubercular lesion, and graft rejection. So when we talk about type 1 of hypersensitivity reaction, we talk also about IgE-mediated hypersensitivity. Type 2, type 3, and type 4, it's a group non-IgE-mediated hypersensitivity. And on this picture, we see mechanism of development allergic reaction, IgE-mediated allergic reaction, or immediate type of allergic reaction. You see antigen recognized by antigen-presenting cell. Antigen-presenting cell present epitope of this antigen on their surface with complex with MHC class 2. And in this case, T lymphocyte can recognize this antigen. After recognition, T helper lymphocyte become T helper 2 lymphocyte and produce different type of cytokines, interleukin 4 and interleukin 10, that activate B lymphocyte. B lymphocyte interact with T lymphocyte and become plasma cell. And this plasma cell produce immunoglobulin E. Immunoglobulin E. After this, immunoglobulin E bind to mast cell on the surface of mast cell, and after re-exposure of antigen, antigen bind with both immunoglobulin E on the surface of mast cell, and we see production of preformed and newly formed mediators from mast cell. And these mediators, histamine, prostaglandins, and leukotrienes, lead to development clinical effect of allergic disease. Which types of allergic disease we have, and what about we will talk in our lectures and in our classes. First of all, common allergic manifestation include allergic rhinitis, allergic conjunctivitis, asthma, atopic dermatitis, urticaria, reaction to food, drug, and also very severe reaction, it's anaphylaxis and anaphylactic shock. On this picture, you see patient with high fever or seasonal allergic rhinitis. High fever, also known as seasonal allergies, has little to do with high and nothing to do with fever. Farmers coined the phrase at the turn of the century because they saw the same symptoms year after year during hives season. Allergic rhinitis characterized by congestion, itching, and dripping of the nose. Next, clinical manifestation of allergy is allergic conjunctivitis. Very often we see development allergic rhinitis and allergic conjunctivitis in the same person. Clinical manifestation or symptoms include redness, itching, and swelling of eyes. Also, in patients with allergic rhinitis, we see allergic sinusitis and allergic otitis. Bronchial asthma. And you know that asthma is a disease affecting the airway. Symptoms may be very different and from person to person, include coughing, chest tightness, shortness of breath, wheezing, and some other symptoms. It's most likely occur in children by age 5 and also in adults in their 30s. Certain things increase the chances of development asthma. For example, if one of parents have asthma, patient will be more likely to get asthma. And also, that's very important, we see allergic rhinitis in 78% patients with asthma. Next, very important disease, atopic dermatitis. Symptoms include itching, reddening, and flaking or peeling of the skin. More often, symptoms begin in childhood. Dermatitis often precedes other allergic disorders, and over 50% of those with atopic dermatitis also develop asthma. Urticaria, also very important disease, because sometimes we see only urticaria, but sometimes we see urticaria and angioedema in the same person. Urticaria characterized by itching red bumps that can occur in clumps and be either large or small. Hives are often triggered by infections, certain food, and medication. Food allergy. The most common food allergy triggers are the protein cow's milk, eggs, peanuts, wheat, soy, fish, shellfish, and tree nuts. More often we see development of food allergy in children, and in adults we see development of food intolerance. Anaphylaxis is an acute systemic type of allergic reaction. It occurs when a person's immune system has been triggered to recognize substances at a threat to the body. Also, anaphylaxis occurs infrequently. It is life-threatening and can occur at any time. And common causes of anaphylaxis include food, and especially it may be nuts, fruit, fish, spices, drug, first of all, penicillin and anesthetics, aspirin, latex, exercise, and bee or wasp sting. And why are allergies increase? And as you can see, the most supported theory behind the increase of allergies is the hygienic hypothesis. Hygienic hypothesis suggests that living conditions in much of the world might to be clear, and that kinds aren't being exposed to germs that train their immune systems to tell the difference between harmless and harmful irritants. And this theory was first introduced in the late 1980s by David Strachan, a professor of epidemiology. And today this theory is supported by several studies that show that in developed countries where there is better sanitation, water supplies, and children with fewer infections, there is a significant increase in allergy compared to population in less developed area. And also this you can see in this picture. Increase development of allergic reaction and protect response healthy person in not good condition. Increased exposure to endotoxins, a poisonous substance present in certain bacteria that stimulate and educate the immune system, decrease allergic inflammation. The hygienic hypothesis suggests that the immune system must be educated to properly function so it can learn to defend against infection organisms. And there is still more research to be done regarding different lifestyle in developed and undeveloped countries that lead to protection against the development of allergies, as well as an expansion of current research looking at way to prevent the development of allergies. And also, you can see this girl, germ-free, genetic, hygiene environment. And in this case, we see using vaccination, sometimes using antibiotics, Western lifestyle. In this case, we see allergen sensitization and development T helper 2 response and development asthma and allergic disease. This boy that have genetic predisposition and poorly hygienic environment. He have early childhood infection, maybe microbial exposure, older siblings, daycare exposure, farming environment, early exposed dogs and cat, and in this boy we see protective response of immune response, and this is healthy boy without asthma. Also, as you can see, when we talk about development of allergic disease, we must talk about anthropogenic factors such as lifestyle, nutrition, pollutants, urbanization, climate changes. And in this case, you see in human with predisposition factors, susceptibility, maybe age, different hormonal factor, different genetic background. If we see this anthropogenic factor in human lead to development allergic disease. Because environmental factor, climate factor, also very important when we talk about development of this group of disease. So, global climate change also influence on development of allergic disease. Sometimes a controversial issue, global climate changes, is also thought to be in part responsible for increase in allergies, making the matter not only an environmental issue, but also a health issue. And on this picture, you see that this is due to allergen triggers such as ragweed that grows faster and produce more pollen under increased carbon dioxide level. In addition, warmer conditions lead to explanation of allergic trees and also increase the amount of fungal antigen in the air. This theory was supported by data from pollen count trends collected by pollen station. And other effect of climate change include the increase of allergic asthma attacks due to the exacerbation of air pollutions and pollen production in cities that leads to the urban heat island effect, as you can see in this picture. As a result of these environmental changes, high fever or seasonal allergic rhinitis lasts up to 3 weeks longer than it used to, and the further north you live, the longer you have to wait for relief. And also you can see that in city we see 3 to 10 degrees hotter than in rural area. And we see changes in water penetration, in plant transpiration, and in some other factors. And all these factors may lead to increased development of allergic disease. And now talk about allergens. What is allergen? And you remember that we talk about antigen, and allergen is antigen causing allergic disease. Structure of allergen. Most allergens are glycoproteins. Molecular weight may be from 5 to 100 kilodalton. Most around 20 to 40, 60 kilodalton. When we talk about pollen, many pollen allergens are enzymes. A genetically predisposed person can become IG sensitized after several years of inhaling of grass pollen allergen per season. Rare, low molecular weight chemicals are still referred to as allergens for IG antibodies. When we see patient with allergic contact dermatitis, the classical allergens are low molecular weight chemicals, for example, chromium, nickel, and formaldehyde reacting with T cells, and we see development delayed type of allergic reaction, and we see development allergic contact dermatitis in this group of patients. What also we must know about allergen. So, allergen is a substance that can bind to immunoglobulin E antibodies. Which type of allergen we have? Allergen source, whole extract, and allergen components. Allergen source are tissues, particles, foods, organisms that trigger allergic immune response. For example, it may be pollen, dust mites, cats, mold, and peanuts, and some others. What is whole extract? Whole extract are a crude mixture of allergic and non-allergic proteins, carbohydrates, lipids, and other molecules extracted from the allergen source. It's a whole extract. And allergen components. Allergen components are molecules that trigger an allergic response. These single allergens can be isolated from nature allergen source and made using recombinant DNA technology. And allergen components very important when we talk about diagnosis of allergic disease, and also it's very important when we use treatment with allergic disease, especially specific immunotherapy in treatment patient with allergic disease. Almost anything containing proteins can be an allergen source. Each allergen source contains many different allergenic protein, allergen component. Also, we can see that, for example, regolith have 8 different allergenic proteins. Each allergen components commonly have several different epitopes, and one epitope can lead to development one immune response. Epitope is an actual 3-dimensionalization binding site for an antibody. So epitope it's a part of allergen that bind with immunoglobulin E, the specific immunoglobulin E. And on this picture, you can see crystallographic structure of PEL-D1 dimer. It's a allergen of cat allergen. And now talk about types of allergen. All allergen we divided into two groups: major allergen and minor allergen. Major allergen. This is allergen that is recognized by IgE antibodies in over 50% of patients allergic to the allergen source. Minor allergen, an allergen that is recognized by IgE antibodies in under 50% of patients allergic to the allergen source. And this allergen major and minor do not always suggest major and minor clinical relevance. Some allergens can be recognized by more 50% of patients, but bind to a small fraction of the immunoglobulin E antibodies. However, in most cases, the major allergen do bind to a large fraction of overall IgE and are often of dominant clinical importance. On this picture, you can see regular antigen, first antigen MA1. It's a very unstable protein. This allergen non-glycosylated, and we have two chains in this allergen. Also, you can see red and yellow chain. And on this picture, you can see alpha and beta chain, and both these chains are reactive with immunoglobulin E. And also, when we talk about types of allergen, we must remember that may be sensitized or primary allergen and non-sensitizing or secondary allergen. Primary allergen. It's an allergen or allergen source that is able to induce allergen-specific immunoglobulin E antibodies. Non-sensitizing or secondary allergen. It's allergen that can only cause allergic symptoms if previous contact with related allergen has caused sensitization. And in this case, we talk about cross-reactivity. Cross-reactivity also very important when we talk about elimination of different type of allergen. And also must know about stability of allergen. First, it's acid pH. Susceptible to acid pH, not able to cross gastric barriers. And also this except possibly in patients treated with antacid drugs. Temperature, also very important. Temperature susceptibility does not maintain its allergenicity after cooking, heating procedures. Temperature susceptibility very important when we talk about food allergy. For example, allergic reaction to milk. Milk have four main very important allergen. Alpha-lactoprotein, very non-stable protein, and after cooking our patient with allergic reaction to this protein can use this milk or milk product. Or casein. Casein, it's stable antigen. And after cooking, patient with allergic reaction to casein can't use this product because we see that this protein contain this product, food product, and we see development of allergic reaction after use cooking product in this group of patients. Why protein stability? Very important when we talk about allergen components. Allergen that's stable to heat and digestion are more likely cause severe reaction. And allergens that are heat and digestion label are more likely to be tolerated or only cause mild or local symptoms. And also we can talk about primary allergen and secondary allergen. Also we remember that secondary allergen lead to development cross-reactivity. What is cross-reactivity and why it is very important for us? Cross-reactivity is a phenomenon of an IgE antibody recognizing, binding, and inducing an immune response to similar allergenic molecules present in different species. For example, it's a reaction between beech pollen and hazelnut because we see in allergen of beech pollen pear 10 and allergen, and also this allergen we see in hazelnut, and our immune system will react on this same antigen. On this picture we see maggot pollen allergens, and you see that this pollen have 6 different types of allergens: Art1, 2, 3, 4, 5, and 6. And Art4, it's prophyllin. It's cross-reactive protein. And when we see regular pollen allergen, also we see the same protein, prophyllin. And so maggot and regular have the same protein, prophyllin. So we see cross-reactivity between this group two, two group of pollen. And next we talk about nomenclature of allergen. World Health Organization and International Union of Immunological Species, using their Latin family name, genus and species. For example, Arachis, Hypogea, Ara H, or peanut. Also, we use a number, distinguished various allergens from the same species, for example, 1, 2, etc. And the numbers are assigned to the allergens in the order of their identification. Allergenic molecules are classified into protein families according to their structure and biological function. And you can see nomenclature of all allergen on www.allergen.org. On this picture, you can see Lolium pyrenea or ryegrass, and nomenclature of Lolium pyrenea, LOL, first 3 letter P, and this is first allergen, LOL-P1, first pollen allergen. Next picture, we see Bermuda grass, Latinum abbreviation Cynodon dactylum. We use first 3 letter C. and then first letter D, and first allergen of Bermuda grass will be CND1. But when we talk about some allergen, for example, domestic dog, Canis domesticus, and mold, Candida albicans, if we use only first three letters, it will be the similar can and can. To avoid ambiguity, the modified system is used to designate can D for domestic dog, Canis domesticus, and can A for Candida albicans, allergens from these two sources respectively. On this picture, you can see birch pollen, and you see pollen grass and major allergen of pollen grass. Betula varicosa 1 allergen, its major allergen, BETV1. And now talk about allergen and either allergen. What's the difference? An allergen from a single species may consist of several closely similar molecules. These similar molecules are designated as either allergens when they share the following common biochemical properties: similar molecular size, identical biological function, if known, for example, enzymatic action, and more than 67% identity of amino acid sequences. And for example, also, we see rugged allergens, MA1 and MA2. They share 65% amino acid sequences identity. These allergens were assigned to different groups because of their different immunochemical properties before their sequences were known. Allergens from different species of the same or different genus which share the above-mentioned common biochemical properties are considered to belong to the same group, and the sequence's identity requirement can be less than 67%, as in the case for allergens of the same species. On this picture, you can see ragweed, Kentucky bluegrass, and ryegrass. The minor pollen allergens MA10 or P10 and low P10 according to nomenclature. Also, their sequences are not known. They are assigned to be the same allergen group as they clearly have the same biological function of cytochrome C. And now we talk about different group of allergens. First of all, we talk about indoor allergens. When we talk about indoor allergens, very important, the most important allergens are domestic house dust mites, animal allergens, cockroach allergens, and fungi. On this picture, you see house dust mites. House dust mites are a common cause of asthma and allergic symptoms. Also, you see major allergen of house dust mites, DERP1. And when we talk about house dust mites, we have two very important classes of dust mites. Dermatophagoides teranissimus in European countries and Dermatophagoides farina in America. Domestic mite allergens are present in various parts of mite bodies, secreta and excreta, and constitute the main source of dust-derived allergens. Domestic mites feed on human and animal scales, and conditions for growth are temperature between 22 and 26 degrees and relative humidity greater than 55%. And very important such species of domestic mites: Dermatophagoides tyrannissimus, Dermatophagoides farina, Dermatophagoides microceras, and Hieroglyphus maina. Dermatophagoides tyrannissimus is the dominant mite in constantly damp climates: Northern Europe, Brazil, and the Pacific Northwest. Dermatophagoides farina survive better in dried climates and in areas with prolonged dry winter. Blomia tropicals commonly found in houses in tropical and subtropical areas. When we talk about allergen of domestic mites, they belong to three groups of proteins: cysteine proteases, group 1 allergens, Dermatophagoides tyrannissimus, Dermatophagoides farina, and Dermatophagoides microcea; serine proteases, group 3 allergens, amylase, group 4 allergens. These allergen enzymes have been found in mice fecal. The predominant allergens in house dust mites are from group 1 and 3, and very little group 2 allergen. And if in house we see concentration of mites allergen above 0.5 milligram of Dermatophagoides tyrannissimus per gram of dust, it's a very significant risk factor for development bronchial asthma. And next group of allergen, it's a pet allergen. And in this picture, you see dog and cat, and also major dog allergen, CAN-F1, and major cat allergen, PEL-D1. And we begin talk about cat, because it's very important allergen. Cat allergens are potent airway sensitizing agent. This allergen easily become airborne because this very small allergen, about 3 to 4 microns in diameter. And main allergen protein PEL-D1 is found in cat pelt, especially facial area, sebaceous secretion, urine, and saliva. Dog allergen. Dog produce two major allergens, CAN-F1 and CAN-F2. And when we talk about characteristic of this allergen, this characteristic very similar to cat allergen. But signs of this allergen more than signs of cat allergen. So we can see sensitization to dog only after direct contact with dog. And allergic sensitivity to dogs is not as common as sensitivity to other mammals. And dog allergens we see in dog hair and danger. Also important, rodent allergen, because many children, many people keep rodent as pets. And the allergenicity of rodent antigens is well known in animal handles who become sensitized to urinary protein. And also, you know this group of allergen, cockroach allergen. Most species of cockroaches live in tropical climates, but in our climates also we see cockroaches. The most common species are American cockroaches, Periplaneta americana, German cockroaches, Blatella germanica, Asian cockroaches, Blatella orientalis, Australian cockroaches, Periplaneta australazea, and brown-banded cockroach, Suppella supuledium. On this picture, you see American cockroaches. And allergens from the German and American cockroaches have been characterized, and their presence in house dust can be measured by using specific monoclonal antibodies. Next group of allergen is fungi. Most and yeast can act as indoor airborne allergen. And in dark, humid, and poorly ventilated areas, it's optimal for indoor fungal growth. Fungi grow well within the systems used for cooling, heating, and humidification. The most common indoor fungal are Penicillium, Aspergillus, Alternaria, Cladosporium, and Candida. And the next group of allergens, this is outdoor allergens. Outdoor allergens include pollens and fungi. On this picture, you can see pollen grass. It's very beautiful and very different. How we identified pollen? Pollen grasses, difficult to distinguish morphologically. On this picture, you can see photo of pollen grass. Most of grasses spherical to ovoidal. Size range from 20 to 110 micrometers. And on this picture also we can see grass pollen. This is pine pollen. Size of this pine pollen from 50 to 100 micrometers with 2 bladders or wings. This is cedar pollen, size 25 to 35 micrometers with thick interior and stellate cytoplasmic contents. On this picture, you can see birch pollen, also diameter from 30 to 45, rounded, triangular, polygonal, and 3 tetra and periparate with onset. And on this picture, also you can see acacia pollen, diameter 50. And also you see roundly quadrangular grains. On this picture, we can see saltbush pollen. This is ambrosia, spheroidal redwood pollen, very important in our country. And when we talk about pollen, we must know about very important scientist. This is Swiss molecular biologist Martin Augerli. He used a scanning electron microscope to look at grains of pollen. And this biologist, Martin Agerli, it's micronaut. They see pollen. And 3D images of pollen are originally black and white, and then enhanced with color look oddly aligned. And on this picture, you can see the smallest pollen in the world. It's a forget-me-not, pollen and plants. On this picture, we can see blowing willow and pollen of this willow. In this picture, very beautiful persil silk tree and pollen of persil silk tree. Also, you can see very beautiful Indian mallow and pollen of Indian mallow. Very important for allergic person, pine tree. And we see pollination of pine tree in early spring. And pollen of pine tree, also very beautiful. And talk about pollen. Pollen. Allergens come mainly from trees, grasses, and weeds. So we divided pollen allergen into three groups: trees pollen, grasses pollen, and weeds pollen, because all of these have different period of pollination. The concentration of pollen varies with location and atmospheric condition. In early spring, we see pollination of trees, grass pollens in the late spring and summer, and weed pollens during summer and fall. On this picture, we see the sum of calendar of pollination. Spring period, season from March to June, we see pollination of trees. Next period, in our country, it's June, and first part of July, it's pollination of grasses. And season August to November, it's period of pollination weeds. On this picture, you can see pollen calendar. Its concentration levels of airborne pollens, which are known cause of allergies. And you see concentration. Green if we see pollen concentration more than 30. And this period very important. You can see Betulia, large concentration, usually it's May, very important, very increased concentration. But period of pollination from middle of April to first day of July. Grasses, period of pollination from June to September in some areas. In our countries, only June to first part of July. And you see the very increased concentration pollen in air in July and August. And outdoor fungal allergen. Also next group of allergen, also very important. Which group of fungal allergen we see outdoor? It's Alternaria and Cladosporium. Alternaria and Cladosporium. And also this fungal risk factor for development of asthma. Fungi tend to be seasonal allergens in temperature zone, where some fungi sporulate on warm, dry summer days and others prefer the rainy nights of fall. And now we talk about prevention of allergy and allergic asthma. Which type of prevention we have? We have primary prevention, secondary prevention, and tertiary prevention. Primary prevention. It's prevention of immunological sensitization. For example, the development of IgE antibodies. Secondary prevention includes preventing the development of an allergic disease following sensitization and especially development of atopic eczema or atopic dermatitis, upper respiratory allergy, and allergic asthma. And certainly prevention and treatment of asthma and allergic disease. And first we talk about primary prevention. What includes primary prevention? First of all, it's avoid smoking. Avoid smoking and exposure to environmental tobacco smoke, particularly during pregnancy. And it's very important, especially in early childhood. And also, we must remember that tobacco smoke should be also removed from workplaces. When we talk about primary prevention, also we must recommend our patient avoid damp housing condition and reduce indoor air pollutants. And in patient with sensitization to different type of allergen as primary prevention, we recommended breastfeeding, especially until 6 months, and no special diet for the lactating mother. Primary prevention also includes eliminate sensitizing and highly irritating agent in occupational environment. If this is not possible, implement methods to prevent employee exposure. Secondary prevention. What includes secondary prevention? First, treat atopic dermatitis topically and possibly with systemic pharmacotherapy to prevent respiratory allergy. Because also we'll talk about that in allergic marsh. First, we see development of atopic dermatitis, then allergic rhinitis, and then allergic asthma. So very important when we see patient, baby with atopic dermatitis, treat this dermatitis to prevent development respiratory rhinitis and asthma. Treat upper airway disease, allergic rhinitis, to reduce the risk development of bronchial asthma. In young children, and if we see already sensitization to house dust mite, pets or cockroaches, specific exposure should be reduced or abolished to prevent onset of allergic disease. And remove employees from occupational exposure if they have developed symptoms caused by occupational allergic sensitization. And tertiary prevention include infant with cow's milk allergy should avoid cow's milk proteins. If a supplement is needed, use hypoallergenic formula if available and affordable to improve symptom control. Patient with allergic asthma, allergic rhinitis, and allergic conjunctivitis, or atopic dermatitis who are allergic to indoor allergens such as dust mite, animal dangers, cockroaches, should eliminate or markedly reduce the exposure to improve symptoms control and prevent exacerbation. So we give recommendation to avoid very important allergen from our door, our patient home. Aim pharmacotherapy, primary towards the underlying inflammatory process. Avoid strictly acetylsalicylic acid or other non-steroidal anti-inflammatory drug in patient who are sensitized to them after an appropriate diagnosis has been confirmed. Also, when we see patient with allergic reaction or non-allergic reaction to non-steroidal anti-inflammatory drug, we give some recommendation about food that also must be avoided in this group of patients because they also contain salicylates, natural salicylates, and may activate exacerbation of disease. And education. Education very important for our patient because patient education regarding precipitation of asthma, allergic symptoms, and especially anaphylaxis is essential. Guided self-management to prevent, assess, and treat symptoms is a key to optimize disease control. And first of all, we talk about patients with anaphylaxis and anaphylactic shock, because after education, we see good result more than in 50% of patients with anaphylaxis. And school policies on asthma and anaphylaxis management are useful. Thank you for your attention and have a nice day.